So what you have in front of you is mostly right. There's some changes, and I'll point them out, and you're going to have to like scratch things out as you go. It's readable. I grant you that it's smaller print, smallish print, but it is readable if you get close to it, and it's a good source. It's a good example to have. On your exam on Wednesday, you can expect to see a question like this one, at least one, possibly two. And if you don't understand what's going on, you're not going to be ready for it. All right. Here's this corn that we've already done. You've already turned in the data from collecting it, right? Is that correct?
Yes.
You had the corn, you had the kernels, yes? You were absent. When you're absent, what should you be doing? You should be watching the video of that day's lesson. Did you watch it? Then you did the lab, because I emailed you the lab as well. When I Jupiter grade message you stuff, and I send you videos of the class material, and you're absent, you had a whole weekend to get it ready, I expect you to spend some time going through that. So you got some work to do yet. And the exam is coming up, so get to moving on that. So as we look at this corn, I want to remind you that when you see a corn kernel or a grain of corn, just like a rice grain, a kernel grain, a corn grain or corn kernel is an embryo. It's an embryo, analogous to a baby. You can take any one of these and plant them, and they will turn into corn plants. They're individual corn plants. There are many different kinds of embryos here. Do you agree? Yeah. Each corn cob has 100 embryos, 100 babies. Each corn cob, there's many corn cobs per plant, so one corn plant can lead to many, 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 many embryos, many babies. Yes?
So each one of them is a baby.
Each one of these is like a human baby, right? An embryo. It's ready to go. Just plant it and it'll grow under the right conditions. Are we clear? So this is after fertilization, after the two haploids became diploid, right? Corn's a little more complicated, but we don't have to get into the genetics of corn. The bottom line is that this corn cob, these different colors are either dominant or they're recessive. The color and whether they're smooth or shrunken is dominant or recessive. By the way, you will have to upload these notes in Cornell Notes. So I see some of you not writing. I'm going to expect you to be, there will be an assignment. There will be expected of you. It'll be called classwork. There's no excuse for anyone not getting that done, right? So either purple smooth, you can write on that. And then you could also add some Cornell notes on the side, right? So you have purple smooth, you have purple wrinkled, you have yellow smooth and yellow wrinkled. And so you see that purple is dominant, right? I would write this down. Purple is on a separate sheet of paper. You have it there for your reference. It's there for you. You can highlight it in your notes. But I would write it down in my notes on a separate sheet of paper in Cornell Notes. That P is for purple, big P is purple, little p is yellow. So how do you get yellow?
Two little p's.
Thank you very much, excellent. Does everybody understand that? Mm-hmm. Do you understand that to get the recessive trait, you need two versions of it? If you have a big P and a little p, it's not yellow, it's purple. So smooth is big S, and little s is shrunken. So these are your four characteristics. Obviously, these are the recessive genes, and these are the dominant genes. In order to get the recessive trait, you have to have two copies of the recessive gene. The allele, right? The word allele. If you read this, you don't have to read the whole thing because we're kind of skimming through it, but you can read it at home if you'd like. We're gonna see that there's a table 1, we'll see it in just a second, that's a cross between the two heterozygous parents. Two heterozygous parents. They're telling you that. they're telling you that the two parents that are heterozygous have a bunch of embryos, and this is what we get. Now, these embryos ideally would be a corn cob. These are all the embryos from the two parents. Does that make sense? Mm-hmm. These are the products of this cross. Two heterozygotes, right? Big P, big S, little s crossed with big P, big S, little s gets you this. Okay? Does that make sense? All right. So do you have any questions? Because if you got a question now, it's just going to get worse as we go along. Yeah?
You said, what are you doing to like when you cross?
This corn cob. This is the results. So each of these embryos is a result of a cross between those two parents. So you get a new plant. That plant has all these embryos. That's it. That's the result of that. All right. Are we good? All right. Let's go on. When we do that cross, this is the ratio we would expect, right? So let me ask you to do this, because you need to be able to do this by Wednesday, right? Can you, taking two heterozygotes, which would be big P, big S, little s, crossed with big P, big S, little s. Those two parents, given those two parents, can you take these two individuals and come up with these embryos, or these, gametes? Do it. Do it right now on paper. Show it to me. When I ask you to upload the notes, I want to see you use foil. Use the arrows. Show me. Demonstrate you understand that these two parents, each one of these can produce these many embryos. I want you to do foil. If you don't know how to do foil, then the problem is, let me tell you where the issue is. When a student gets this on a test and they're asked to make something like this, what ends up happening is the student that doesn't know how to use foil will end up putting big P, big S, little s, big little p. They'll put all kinds of options here except for the 4 possible gametes, the 4 possible eggs and the 4 possible pollens, right? Does that make sense? Because what happens is kids don't understand how to create foil, how to create the embryos using foil.
You want us to write down the charts?
You can, but the chart is less important to me than how do you get this row and this column. How do you get the gametes? How do you get the gametes? These are the embryos, right? Does that make sense? These are diploid. They have 2 copies of each gene. These are the gametes. They only have 1 copy of each gene. So how do you get the gametes? Because the people that mess this up are the people that can't make the gametes correctly. They get confused. Why are students confused? Why does this confuse? It's not that you take this P and put it here, this P put it here, this S put it here, this S here. That's easy for people, okay? The hard part is getting this row and this column. They don't know how to make the gametes. How do you get the gametes in the right order? If you don't know how to do that, then you're lost. So I'm asking you to make the gametes right now. I'm going to do it with you in a second. I want you to try to do it on your own. And then compare what you did to what I do and see if you can do it right. Because you have to be able to do it right by Wednesday.
I don't understand what you're saying. I don't understand.
You don't understand that. You don't understand foil. Do you not remember foil?
No.
Do you have algebra right now?
Yeah.
With Miss Simpson or?
I don't understand algebra either.
Okay, but so you got to take it one step at a time. If you absolutely don't understand, then pay extra attention when I explain it in just about 30 seconds. But right now, what I'd like you to do is that you see here that there's 2 copies of each gene. I'd like you to come up with all the combinations of one of each of these that this creature can make, and do the same for this creature. How many different, remember you need 1 P and 1 S, how many different combinations can this creature make, and how many different combinations can this creature make? And I'll go through foil with you in just a minute. Now, the reason the algebra teacher taught you FOIL, and the reason I'm using the word FOIL, is because it's really the best way to get all the possible combinations in a very clear and concise way. So, FOIL, F.O.I.L. First, outer, inner, last. Right? All right. So, what's the first?
It's big P and big S.
Big P and big S. So, big P and big S. See? That's one possible gamete.
Big P and small.
That's right. And so now we go outer, so that's big P and small s. Perfect. Big P and small s. So that's another option. That's another possible gamete.
Little P and big S.
Very good. Then little p and big S, that's the inner.
Little P and small S.
Little p, oh, let me do that again. Little p and big S. And then little p and little s. The four possible gametes that this creature can make. What are the four possible combinations of genes that different combinations of genes?
It's the same thing for the other one.
It's exactly right. It would be the same thing for the other one because it's outer, first, which would be big P, big S, then big P, little s, then little p, big S, and then little p, little s. So you end up with the same thing is right, which is why these four are the same as these four. Because you have big P, big S, big P, little s, little p, big S, and little p, little s. These four and these four are the same because both parents are double heterozygotes. This part is where everybody messes up when you're doing a cross. When you're doing a dihybrid cross. Now, will they ask you to do a dihybrid cross on the end of course exam? I doubt it. But will they ask you to analyze one? Probably.
You said both parents are double what?
Heterozygote. I'm going to show you some examples I already have. We've gone through them last week, but I'll go through them again. Different examples where they're not heterozygote. And you'll see what the difference is between the two. Yes, sir. No, homo means the same, but there's no. How are these the same? Are you saying this one's homozygous, this one? No. Homozygous means the two copies of genes that you have are the same. Do these have two of the same P's? Do they have, are these two genetically identical for these two genes? Yes. Big P, little p, big S, little s. Yes. But homozygous means that your genes, the two copies that you have, are the same.
So like big P, small s.
Exactly. Big P, big S, small s. Those are homozygous. When you have two different individuals, right? They're not homozygous to one another. Homozygous means your chromosomes have the same versions of the alleles. Let's look at a homozygous, shall we? Let's take a look at what happens if one of these are homozygous for just one character. Big P, little p, big S. How many different gametes can this creature make? Do it. Do the FOIL. Do it on paper so you get the practice.
I like the Punisher back before.
Which one? No, what did you say?
I like the Punisher before.
The Punnett square. You can't get the Punnett square here of this dihybrid cross without FOIL. You have to use FOIL to get the possibilities because These are the 4 possibilities because you did FOIL. Now let's do FOIL here. How many different kinds of, only 2. 2, because look, big P can go with big S, but won't the big P with the big S be the same again? So there's only big P, big S for that first option. Now the second option is what? Little p, big S. Those are your only 2 possible gametes. Will you produce 4 eggs? Sure. But you'll either have this or this. You won't have 4 eggs with little p, little s. Why can't you get little p, little s?
Because
There's no little s. There's no little s. This is a homozygous dominant for S. This is homozygous dominant for S. Do you hear that? This creature is homozygous dominant for S. It's heterozygote for the B, for the P. Now what if you had a homozygous for both genes? What if you had big P, big S, big S? How many different kinds of creatures could you make then? Only 1. So understand, and please pay attention, because this is the FOIL again. If you did FOIL, big P goes with big S. If you did, no matter how many different, whether you did inner,
It's the same.
it's the same. It's all big P, big S. Only 1 option. Here you have 2 options. Here you have 4 options. Are we all clear on this?
Yes.
Because you're going to have to do this. All right, so given that all this is nice, but it's not, these are examples. This is what we had to do right here. So we had FOIL. We produced our 4 different gametes, the male and the female gametes, and they produced all these embryos, and the embryos are here, right? Let's go ahead and count how many different kinds we had. By the way, this is like the 3rd time we've looked at a 9:3 to 3:1 ratio. So you should be expecting a 9:3 to 3:1 ratio problem on your test, right? Okay. So it says 4 different phenotypes which define fractional ratios, 9:16 purple and smooth. Well, let's make those blue. Let's make those blue. 9/16 purple and smooth. This was purple and smooth. Why? Because you have a big P and a big S. All you need is 1 big P and 1 big S. Right? That's all you need to have purple and smooth. This is 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 purple and smooth. They don't have the same genotype, but this one's not purple and smooth. Well, the next one's what? Purple and what? Shrunken or shriveled, right? Why? Because there's 2 little S's. This is purple and smooth. That's not. This is purple and smooth. This is purple and smooth. This is purple and smooth. So how many is that? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9. Right, that's good. But I just want to make sure you see it. That the pattern is the same. No matter what the genes are, the pattern's always going to be the same, assuming you have what? 2 heterozygote parents that give you these gametes. This combination is always going to end up the same, which is why we have you memorize 9:3 to 3:1. All right, so the next one is red. Let's see, it says purple and shrunken. So this is purple and shrunken, right? This is purple and shrunken. This is purple and shrunken. And those are the only ones that are purple and shrunken. Do you agree?
Yeah.
Okay. So there's 3 out of 16. Yellow and smooth. Yellow and smooth would be yellow and smooth, yellow and smooth, yellow and smooth. Again, 3 out of 16. And the last one is going to be yellow and shrunken, and yellow and shrunken, and we're going to color it pink. Yellow and shrunken then is just this last one here, this double homozygote recessive. Are we getting that? Because there was a question over here about what was homozygous, what is heterozygous. This is 2 homozygous genes here. This is homozygous for P, but heterozygote for S. These are both homozygous. This is homozygous dominant for both. This is homozygous recessive for both. Right? And let me tell you right now, I'm going to say it plainly. The key to every genetics problem, almost every genetic problem you're going to have, the very key, the foundation that's gonna help you solve almost any genetic problem. Are you listening? Hello, Mr. I'm gonna lay my head down and not pay attention. Tap him on the shoulder.
Charles.
Yo, sit up, sit up. Why are you rolling your eyes at me? I don't understand. So, whatever. The bottom line is this, the key to solving all these problems is your homozygous individuals. Homozygous recessive is the key. If you have a homozygous recessive, you know exactly what it is. All you have to know, if you have a yellow and shrunken, you know what their genes are, right? If you have yellow shrunken, you know it's little p, little p, little s, little s. Guaranteed. Why? God bless you. That's right. You only get recessive with the recessive genes. You only get the recessive trait with the recessive genes. So if you have to have little p, little s, little s. However, if you have homozygous dominant, how do you know it's homozygous dominant and not heterozygote? You don't. Because you have, in both cases, you have a P. Look at this. Would you see a difference between these two? No, you would not. They're both purple and smooth. Right? Even though this is a heterozygote for smooth and this is a homozygous for smooth, you wouldn't know. So the key to all these problems is this homozygous recessive. Of course, if you did the genotypic ratio, this is what you get, but it doesn't help us too much when we discuss that. Yes?
I think you should give like a worksheet with like the bunch of them on there. And then we should like either homozygous recessive or homozygous dominant or heterozygous.
Why don't we start first with doing this and making sure we understand each other and know what we're talking about. Taking notes, writing the notes down, studying our notes instead of asking for worksheets, right? Are you going to get worksheets? Sure. Are you ready for them? I don't know. I got people here that don't understand the difference. That just now, after a week of talking about this and doing problems, by the way, and doing a lab, still don't understand the difference between homozygote and heterozygote. Some people don't know FOIL, even though they covered it in algebra and here in this class.
You just said
it. Easy. We're not. No one's talking about anyone specific. This is about the fact: are you ready to do your worksheet on your own? I don't think so. Yeah. Question? Yes, you may. So what are the different number of phenotypes is 2 to the second power would equal 4. So you have 2 different genes, a P and an S, right? With 2 alleles each, you get 4 different phenotypes. What if you had 3 genes with 2 alleles each? You can get how many different gametes? 9 different gametes. So as you add genes with a dominant, with more than one allele, you start to see an exponential growth of how many different gametes you can have. Now take that number, put the number 10,000 there, and put 2, 3, 4 different, like for instance, blood type in humans has 4 different options, right? Can you imagine how many different gametes you could possibly have? And then you're recombining them, and then you're having sex, so you're having 2 individuals who make all those different gametes that can then recombine. That's why each human being is so individual. That's why you never have more than one person with your particular combination of genes, unless you have a twin, an identical twin. And even then, because of epigenetics and changes and impacts of the environment, your particular combination of genes is never the same. All right, let's take a look at a problem. This is the kind of problem you're going to have. Go ahead and write this out. Given which is dominant, which is recessive, go ahead and write it out. Make a little chart in your notes of what is given to you here. Remember that big P is purple, little p is yellow, big S is smooth, and little s is shrunken. Go ahead and write it out. I'll pause. pause here and then we'll come back when I've given you a couple minutes. This is your practice. You wanted a worksheet? This is your practice right now. Do it. Write down in a chart. This information is giving you a phenotype. Write out what it is, the genotype, given this phenotype. Go. Okay, so let's go ahead and do this. Now that some people have tried and others have wasted time, let's go ahead and do this together. So we have 381 grains total. Now what does that mean, total grains? Remember, what that means is that there's 381 corn kernels that we counted on a cob, right? Does that make sense? 381 of them. 381 embryos,
Yes.
right? 381, not gametes, embryos. All right.
You divide that by 2.
Of those 216, let's go ahead and write it down, 216 are purple and smooth. Purple and smooth. So what's the genotype for purple and smooth? Big P, and the other one can be either little or big, couldn't it? So either you can do it right at one of two ways. You can do big P, little p, or big P, big P. I think that's a waste of space, and it gets confusing after a while. So what I would suggest is you write big P blank, or big P question mark, whatever you want, because it could be either one. But we know there's at least one big P because it's purple, right?
Yeah.
All right, and what's the S? Smooth, so that's what? Big S blank.
Yes.
All right, so it could be, again, either big S, or big S, little s. We don't know. This is why your recessive is so important. So that's this one. (216) purple and smooth. The next is 79 purple and shrunken. So now 79 are purple and shrunken. So purple's what? Big P, blank, and then shrunken? Little s, little s. There's that homozygous recessive. It's always so important because it just tells you exactly what the genotype is. There's no question it's little s, little s. How do I know that? Because it's shrunken, and shrunken's recessive. The next 65 yellow and smooth. 65 yellow and smooth. Yellow is what? Little p, and smooth is what? Big S blank. We're not sure what that is. It could be another big S, it could be a little s, we're not sure. And our favorite child that gives us all the information in the world is this 21, which is what? Little p, little s, little s. We know these 21, we know exactly what their genotype is. That's what's so wonderful about these homozygous recessives. And you'll see when we do a test cross how important they are. They give us all the information in the world. They allow us to solve all kinds of complicated genetic problems. All right. Sorry about that. All right, so your hypothesis is that you have this ear corn, ear meaning the corn cob, and it's produced by a dihybrid cross. Your hypothesis is that this cross was between two heterozygotes. So two pairs of heterozygote genes, resulting in a theoretical expected ratio of what? 9 to 3 to 3 to 1. Right? You saw the table, some people wanted to make it, so that's what we expect. Well, how do you know? How do you know? This doesn't say 9 to 3 to 3 to 1, does it? It says 216, 79, 65, and 21. That's not 9 to 3 to 3 to 1. Say that again? How are we going to get this? So everybody wrote this down, because if you don't have this written down, when we go to the next page, you're not going to be able to discuss it. So make sure you have that. It's this part right here that you need, right? All right. So let's move on. This is the this is the graph that this is what you're going to have to be able to do chi-square. How do we know that this data, that these results actually prove that this stuff, that these genes are in fact the result, that these results are the result of a cross between two heterozygotes? How do we know that this is a dominant recessive inheritance at all? How do we know that there's not some other kind of genetics happening that we haven't discussed yet or discovered yet? Well, we're never for sure, you're right, but there is some evidence in chi-square. Chi-square is used everywhere, by the way. It's used in business, it's used in law, it's used in obviously in any kind of statistics. It's a statistical analysis that lets you know how closely do your observed results, how close are they to your expected results? That's what chi-square is. So to remind you what chi-square is, let me write down the equation. You wrote it down last week. Had you done your job in Cornell Notes, you would have written it down and studied it, and you would be able to tell it to me right now. What is chi-square? Chi-square is what? What's the formula? You spell it chi-square. What is chi-square? What does it equal? It's the sum of the observed minus the expected squared divided by the expected. It's the observed, what you actually see when you do the experiment. What is it that you see? Or when you count how many car crashes are teenagers involved with, how many did you expect there to be? And is there a difference? Is there something to the idea that teenagers get into more car accidents than older drivers? So we can do what we do when we have that question, any kind of question, is we collect data. We count how many accidents are there. We count how many accidents do teenagers get involved with. And we look at how many we expected, and we do this calculation. And it tells us if the number that we expected is really far away from what we observe, then maybe our theory's wrong. There's something wrong with our hypothesis. This is testing your hypothesis. It's figuring out how close are your actual results to what you see. So obviously it makes sense that we involve both the observed and the expected. So how close are your observed to your expected? Well, you subtract the two, and that tells you the difference between the two, right? And the reason we square it is so that we get no negative numbers. And we divide it by our expected, so that gives us a number that's like an average. Like let's just say it gives us some kind of relationship, a ratio, right? That ratio then, we can add them all up and come up with chi-square. That's what this symbol means, sum, the sum of. That's what this means, the sum of. Anytime you see this in calculus, if you see it in algebra trig, if you see it anywhere you see it in life, in the statistics, if you see this E in math, it means the sum of. We add a bunch of these together. We're going to do this a bunch of times, and we're going to add them together. We're going to do this, in fact, once for every one of our phenotypes. Does that make sense? So then we're going to add those together. So we have our purple smooth. You have this already written down, so you don't have to copy it. Yes, you may. You have purple smooth. You have 216. You observe 216 purple smooth. 216. Now, the way they do the observed ratio is they take, by the way, this is yellow shrunken. Do you see this? This actually belongs up here, okay? This was just cut off on the page. So the way they do this, and you can do it more than one way. You could take, the observed is what you see, right? We already wrote those numbers down. Do you agree? That's where these numbers, 216, 79, 65, 21, we literally just wrote those numbers down here, 216, 79, 65, 21. That's where we got those numbers. Is that clear? We observed them. We counted them on the corn cob. The observed ratio is how many to what? Now you could do a ratio of 216 to the total, which would be 381, or you could do 216 to 21. Either way, it's 216 to 79. Why do we do that? Because one way of doing that, one reason to do that, is we know that 21 should be the number 1, right? Because it's 9 to 3 to 3 to 1, right? Do you agree? So we could say, what is this compared to this 1? So if we take 21 and divide it by 21, what do we get? 1. We take 65 divided by 21, we get what? 3.1. 79 divided by 21, we get 3.8. 216 divided by 21 is 10.3. Because it's 9 to 3 to 3 to 1, right? It simplifies the math. Instead of showing 9 to 16, 216 to 381, what is the ratio, simplifying it, dividing it? This simplifies things. It makes things go quicker, yes? So that's an interesting way of doing it. I had never seen it done that way, but I'm not a statistician, so maybe this is common. So these are your observed numbers. This is your observed ratio. This is what we expect. We expect 9 to 3 to 3 to 1, right? Does that make sense where we get our expected? All right. Please make sure it does, because when we do a regular cross, we expect 3 to 1, for instance. When we do this number, these expected might change depending on the problem you're dealing with. And we'll talk about that later. So know that the expected ratio is, because you know this is a heterozygous cross, you expect to see 9 to 3 to 3 to 1 in the genotype, in the phenotype of the embryos. What do we get? 10.3, 3.8, 3.1, and 1. Are these two close enough?
Yeah.
Is 9 close enough to 10.3? Is 3 close enough to 3.8? Is 3 close enough to 3.1? I don't know. You say yeah. Why do you say yeah? How do you know that means that difference doesn't mean doesn't equate to something being wrong? That this is not dominant recessive. That these are linked or these are not linked. Huh? The answer is you don't know. Yeah, I'm glad. You don't know. And so that's why we do this whole chi-square thing. The reason we're doing all this is to try to figure out, is this close enough or not? When you're done calculating chi-square, you're going to compare it to a table. And that table, that comparison is going to tell you, this is close enough or it's not close enough. If it's not close enough, you have to reject the null hypothesis, and if it's close enough, then you accept the null hypothesis. And we'll talk about what the null hypothesis is in just a second. All right? So, you take your expected, what's your expected number if your expected ratio is 9? It's 9/16 times 381. Where'd you get 381? The total number. There's 214, that's 71. So 71 compared to 79, 71 compared to 65, 24 compared to 21, and 214 compared to 216. That's what you're expecting. Once you have your expected number, you have your observed number, yes? Now you can do this. Observed minus expected squared divided by expected. Where'd I get 4? 4 divided by 214 equals this. Where'd I get 4? Do the math. I see, I hear you. You're right. I can think you're right. If you're paying attention, you should know the answer. Should I make this a quiz? Have you write it down on a piece of paper, turn it in? It's observed minus expected. What's observed? 216 minus expected, which is what? 214, which is what? 216 minus 214 is what? 2. But this is 4. What? Squared. It's this minus this squared. Observed minus expected squared. Divided by expected, so 4 divided by 214 equals this. Let's do the next one together. Understand you're going to have to do this on the exam by yourself. This is subtracting and adding and squaring using a calculator. I don't see this being a problem, assuming you understand what's going on here. Observed minus expected squared divided by expected. Let's do it here. Where did they get 64? Observed minus expected. How many did we expect? Which is how? What's the difference? 8. 8 squared is divided by 71 is this. It's pretty straightforward. Now the next one is 36. Where'd they get 36? 65 minus 71. That's a negative number, but when we square it, it's not negative anymore, right? What's 65 minus 71? 71 minus 65. 6. It would be negative 6 if it's 65 minus 71. Yes, it's negative 6. Negative 6 squared is 36, positive 36. That's why we square it, so we have no negatives involved in this calculation. Divided by 71, because that's the expected. It gives you this number. See, the closer this is to 1, the closer that you are to your expected. If you get a 1 here, if this is a 1, you're very close to your expected. You want to get, it would be very spot on if it was all 1. It almost never happens in real science. Yeah.
So all you gotta do is find the square root of each, like the square root.
The square, not the square root. Just you're squaring
No,
it.
I'm talking about for the observation number, like 4, 64, 30.
If you took the square root of this, what would you get? The square root of 0.05, 0.507?
No, for like 4, 64, 36, to find your difference between your observation number and your expected number.
All you do is take your observed minus your expected.
I know what I'm saying, like, you could just do the square root to find it out.
Square root of what?
Like, yo, what's that? I don't know if that's the observation number.
You mean this number?
No.
Just take the square root of this?
Like 4, 64, them numbers, 36.
Where do you see? Where do you see 464? Oh, these here?
Yeah.
Oh, yeah. Yeah, but you won't know that. You don't know what this is. This is the square, taking a square root of this would just give you the difference between the two.
That's what I said.
So it would be 2. So that would tell you the difference between the two. If you took the square root of this number, this value right here will give you that there's a 2 difference, but you don't know what the observed and the expected would be. You have to have this filled in. It doesn't do you a lot of good to go backwards in this problem. Yeah.
When you get this, where do you get the 16 from? Is that
16 where? Where do you see the 16?
So it's like 9.
Oh, this 16?
Do you just add all of those, like 9 and 33?
9, 3 plus 1 is 16, that's right. And remember, if you did the dihybrid cross, remember that, look at the chart that you have, that I published for you, that I printed for you. If you count the total number of embryos in that dihybrid cross, how many do you think there are?
Wait, which one?
The dihybrid cross.
Oh.
There's 16.
This right here?
I can't see, so you have to bring it around here. Quickly. But if you look at the dihybrid cross, look at how many embryos there are. Remember we highlighted in blue and yellow and that? Yeah, that's it. Count how many you have. 4 by 4, it's 16. 16 different embryos, yeah?
Okay, so in the expected number column 5, how did they get equals to 14 and equals 71?
Okay, so if you take 381, do you have a calculator in front of you? Get a calculator. Use your phone. Do the math right here now. right, so let's do this. 381 times 9, do the math. 381 times 9, and divide that by 16. Tell me what number you get.
Divide by 16 or
Yeah.
9
Well, it's multiplied. See, 381 times 9
over 16.
divided by 16.
Okay, I did that. Okay.
And what'd you get?
214.
Well, 214, yeah? Let's do the next one. 381 times 3. Do that. Divided by 16. What'd you get?
Bless you.
71, yeah? So all it is this number, which is the total, times the fraction that you expect it to be.
Are these gonna be on the test?
What?
Like these problems? Like, are you gonna give us problems?
I literally said that 3 times.
Okay.
You are gonna get a problem like this on the test, yes.
Are we gonna get calculators?
You need to have your own calculator as part of your supply list. Do I have some extras? Maybe. I might have some laying about, but I don't have enough for everybody. You need that. No, you may not use your phone on the test. Are we clear on this? How do we get this?
Yes.
So all you're doing is just taking your total, multiplying times your expected fraction, that gives you your expected number. Then this is just simply a squaring and a subtracting. All right. Let's go on. When chi-square is the sum of all of these, so all of these, 0.019, 0.901, 0.507, 0.375, added together, give you chi-square. Your chi-square value is 1.8. That is chi-square. You figured it out. Once you have summed it all up, you have your chi-square value. But that by itself doesn't give you any information, does it? I got 1.8, yeah! Well, that doesn't tell you anything. You need to compare it to another number. Are you ready? But this is the whole point. This is your chi-square. This value here, chi-square, is a sum, and that's what this means here, sum, is the sum of this all added together. So all these added together give you your chi-square. Is that clear? All right. So let's go on and see what we got. So 1 point step then. So this is chi-square. Oh, I missed something. Our chi-square value. So let me write that down. So your chi-square value is 1.8. Now let's see what we have, something that we have to calculate, and that's our degrees of freedom. You have to calculate your degrees of freedom. This is the simplest thing you'd ever have to do in any math or science class. Your degrees of freedom are the total number of variables, total number of options you have, minus 1. How many different options do we have with these two parents? How many different options do we have? Why do we have 4? You're right, it is 4. What are those 4? Purple and smooth, yellow and smooth, purple and shrunken, yellow and shrunken. Those are our 4 possibilities. The degrees of freedom is your possibilities, which is 4 minus 1. So in this problem, it's 4 minus 1, which is what? 3. So your degrees of freedom are 3. Your degrees of freedom is, in this case, 3. That's what you need. Now, we're going to look at this table. Now that we're going to have to figure out what this probability is. Now that we have our degrees of freedom, now we have to figure out what this is. Now, let's be clear on what this means. In this class, and in most of the time, you're going to be looking at the 0.05 probability. Yeah, you're going to have this big table. In some in some circumstances, somebody might tell you, I want you to use 0.95, somebody might tell you you want 0.2, somebody might tell you you want 0.1. But in this class, and in most classes you take, you're going to assume, I'm sorry, not 0.5. You're going to assume at 0.05, 0.05. You're going to look at 0.05. Is that clear?
So yeah, I heard about it.
Okay, that's all right, that's all right. So it's 0.05 is what's significant or not. Do you see where it says it's significant? So really, this is where we want to stick to. At a 0.05, if there's a difference, so what does this mean at 0.05? What does this chi-square mean, and what do we do? We're going to have to wait out for that tomorrow, okay? We're going to talk about that tomorrow. So read the rest of this tonight. It's going to be a little complicated, but I think you'll see it'll make sense tomorrow. All right? Okay. So the null hypothesis, N-U-L-L, hypothesis, is the hypothesis that states quite simply that any difference between the expected and the observed is due to randomness. Due to random events. So to be clear, that means that, another way of saying it, there is no significant difference. So there's going to be a difference, right? We don't expect it to be matching exactly. We expect that there is going to be some difference because of human measurement error, because of random events that we can't predict. We expect to see there to be some difference from what we predict. Predictions are not 100% correct. In fact, one of the things in science that's a big flag, if you're a scientist reading someone's journal, one of the things that says, uh-oh, somebody might be cheating, is when things match up too well, when there is no error. Then you're just thinking, that's not right. Because that's not the real world. Yeah?
So like, if you do the Chisquare and your values get close to the 9.33 to 1 ratio, that would be the right acceptance?
It's a really great question. So to apply this null hypothesis, it would be, that's exactly right. If your 9 to 3 to 3 to 1 ratio is what you expect, if your results are very, very close to that, then you just say, hey, it's not, the difference is not significant, right? That means that any difference is just due to randomness. So what I do is I accept the null hypothesis, right? I accept the, so in other words, there is nothing else going on. It's exactly the way that I expected. If on the other hand, there is a big difference, then I reject the null hypothesis and I have to say something else is going on. Does that make sense? Let's take a look at this here. So there, in this chart that you have a copy of, there is this value. This number here is called the p-value. All right, it's p, the probability value. You can have the lower that number, and most of us, most of the time, you're going to see people picking 0.05. 0.05. All right. So if you pick 0.01, then the numbers are going to be highly significant. If you pick 0.05, they're still significant. You can pick any one of these probability numbers, all right? So when you do chi-square, if your number is these values here, I should point this out here. These values here, these are the chi-square values. So this is what we call critical value. This is what you're going to compare your chi-square value to. So these are your critical value numbers. Any chi-square problem I give you, you can use that chart. All right? Don't write anything else on that paper, but you're going to be able to use that chart. So if your chi-square ends up being 1.80, and you're using this p-value of 0.05, you're going to use that value. So this is the column you're going to use. And you have a degree of freedom of 3. And remember, what's the degree of freedom mean? How many options there are? -1. So there's 4 options, -1 is 3. So what you're gonna do is you're gonna use this column, this row right here. And of course you're using this column because we just said we're gonna use 0.05. In general, in this class, we're always gonna use 0.05. So with the critical value then, with 3 degrees of freedom and 0.05 significance, the critical value is 7.82. All right? 7.82. That's what you're going to compare your chi-square to. And you can read this here, and it tells you everything you need to know, I think, about chi-square goodness of fit. So if you read it, one of the things you'll see is it says, another approach to arrive on the same conclusions compared to the calculated value of chi-square, the table 0.05 for a given degree of freedom. If the calculated value is less than the table value, the goodness of fit of the given ratio is accepted, otherwise it's rejected. So if the calculated value is less than the calculated value of your chi-square is what? Where is that? 1.80. If that number of chi-square that you just calculated yesterday, if it's less than the critical value, then what do? Then you accept that the original hypothesis is correct. You accept the null that any difference between the two, you're accepting the null hypothesis. You're saying, listen, any difference between what I expected is just due to random luck. If the chi-square value is bigger than 7.82, then you're saying, wait, the difference is too big. It can't be due to random luck. Something else is going on here. Experimental error, linked genes, different ways of inheriting in the case of genetics. I don't know. Something else is going on. Yeah, question?
So you said if the calculation is less than what?
Less than the critical value. What's the critical value? In this case, 7.82, and the p-value in this column. We're looking at this column at this degree of freedom. Did you see that?
So if it's less, you accept the null hypothesis?
If it's less than this number, then you accept the null hypothesis, which is saying that the difference is not significant. If it's more than this, if it's equal to or more than this, then you're like, whoa, this is different. There's something else going on here. Yeah.
So it wouldn't be the other way around, because like, say our hypothesis was that we were crossing 2 heterozygous. So if the results were close to 93:1 ratio, wouldn't that accept our hypothesis?
All right, say that again.
Like, wouldn't it be the other way around? Because, like, say our hypothesis was that we are crossing 2 heterozygous.
All right, so we're saying that we are crossing. Think about what chi-square is. So let's think about it. Let's write out the formula for chi-square, okay? So chi-square is equal to the observed minus the expected squared divided by the expected. and we're going to sum it up for every option, all the data points, right? And we're saying that that difference is a number, some number. So what are we doing when we calculate this? We're looking at the observed, and we're looking at the difference between the observed and the expected. When that number is really big, when the observed and the expected are very big, if the difference between them is very, very large, the bigger this is, the less likely that the observed is close to the expected. Is that right? Do you agree with that? So the bigger the chi-square is, the less likely that you're going to get, that you're anywhere near what you should be getting, that your hypothesis is correct. So we don't really try to think of it as a hypothesis. We're testing the null hypothesis. So when we do chi-square, what we test is the null hypothesis. What does the null hypothesis say? That any difference between these two, whatever the difference is, any difference is due to what? Random events. So if we reject this, if we reject the null hypothesis, what are we saying? There's a significant difference. There's something else is going on. It's not whatever it is we think it is. If we think it, if it's very, very close, in this case it's 1.80, it's very close. In fact, if we did it the way the book, the original document that we looked at yesterday gives you, 1.80, if we looked at 3 degrees of freedom and found 1.80, it would be somewhere around here, around 60%. So you're about 60% sure that it's two heterozygous and aren't linked. Does that make sense? But in science, we actually accept 0.05, a 5% significant difference. So that's the key, is that this is actually very, very, a 1.80 is very good number, and it's acceptable. It's all well within experimental error. Are we okay with this idea? Significance does not mean cause and effect. This does not prove that it is, right? The only thing it says is that our data does not disprove it. Does that make sense to you? Because can we ever prove anything in science? No, we can't. All we could ever do is work towards disproving it. So as far as we know, the evidence that we have so far collected, these two genes are not linked. These two individuals were heterozygote for both genes, and the results were as we expected, close enough to what we expected that we have no evidence to suggest that there's anything else going on.
So we reject the null hypothesis?
In this case, we're accepting the null hypothesis. We're rejecting the original hypothesis. We're rejecting the null hypothesis, which means what?
That our hypothesis is
We're accepting the null hypothesis in this case, so there's no significant difference, right? There is no significant difference. The hypothesis would be there is a significant difference, right? So that means that we're wrong. But if you accept the null hypothesis, then there is no significant difference from expected. Are we good? Yes or no? Okay, so...
No, wait. If that's 7, that's 7.82,
and if the chi-square is 1.8 and the critical value is 7.82,
that's different.
is 1.8 bigger or smaller than 7.82? So we accept the null hypothesis that there is no difference, that the difference between, which is 1.8, it's not 0, right? 1.8 is not 0. It's not exactly on. There is a difference, but it's just not significant. It doesn't. Anything that it's well within if it was all the way up to 7.82, we could still say it's due to random error. So 1.8 is well within, a value that's acceptable. Are we good with this idea? Yes or no? Yeah.
I think the problem that I'm having is actually solving to find the values.
Well, okay, well, let's go back. I'll stop here and then we'll look at the video and we'll walk through it one more time and then I'm gonna give you some sample problems. In your, actually, you only turn half the lights on so that people can still see up here. So, in this practice problem, they tell you you have 1,600 cards, right? And they tell you that you have spades, and you have hearts, and you have diamonds, and you have clubs. And these are your observed ratios. What you see of your observed numbers are 404, 420, 400, and 376. That's what you see. My question to you is, what do you expect? You have 1,600 cards.
Well, I see even.
For all to be equal.
What would you expect to see? That all of them would have 400, right? A couple people said it, and I think it's reasonable. So your expected would be this, right? That's all you need to get chi-square going, right? You're ready to go. What are you asking? Is this reasonable? If this is what I get, is someone cheating, or is this reasonable?
No,
somebody cheated.
Well, these are, you got to admit, you have to say that again.
24, never mind.
All right, so this is all 1,600. This equals 1,600. So the question is this casino cheating me, or do I actually? Is this a reasonable is this well within random luck that we get this distribution instead of this distribution?
No.
Well, we have to figure it out. What are we going to do? Chi-square. I'm going to remind you what chi-square is. Chi-square is equal to the square of the observed minus the expected squared divided by the expected, but that's not all, right? It's right. It's the sum of it's the sum of that, right? So it's the sum of this expression. So what you're gonna do is hopefully you figured this out. You're gonna do a chi-square area, and you're gonna calculate the chi-square for each one, and then you're gonna do what? After you've calculated those numbers, what are you gonna do? Add them. This means the sum of. What's your degrees of freedom here? 3. It's 4 minus 1. It's not hard, is it? Now, yeah, someone can. So the second one, though, that's the first problem. Everybody should be able to do that one easily. The second problem is a little more difficult. The second problem asks you to look at the same situation, except what? You got jokers. Same situation plus jokers. Remember, for every 52 cards, you have 2 jokers? All right? So you got some ratios there to figure out. You guys can figure this out, though. I'm sure I have all confidence in you. And you should be able to figure that out. And then the third problem is a genetics issue. Is a genetic engineer, has this many phenotypes. This is what he was expecting. Is his expectation merited? Does his model fit the data? And then the last experiment is a standard dihybrid cross, again doing chi-square. So you guys are on your own the rest of the period. You can work in groups if you'd like, but get it done. With what I gave you there. If for problem 2, you're adding jokers, and that's an issue. I sent you a message. I made a mistake when I wrote that. I said if you have 52 cards, you expect to see 2 jokers per deck. That's not right. There's 52 cards, and then you add 2 jokers, it's 54 cards in a deck. When you include the jokers, there's 54 cards. So your total's 54. So do you want me to walk you through how to begin that one, or do you want to try to handle that on your own?
Please walk me through it.
You guys are ridiculous. All right.
Can you move the test back Thursday?
Could you just, could you listen?
Yeah, I'm gonna listen.
All right. So, if you take a look at what you got there, you got a list of spades, right? Just like you did before, hearts, and you have diamonds, and you have clubs, and you have jokers. And what you observe when you lay out the deck is 404, 420, 400, 356, and then 82. That's what you observe. But what did you expect? By the way, if you add all those together, you get 1,662. Is that right? Okay. That's what you get. And if you have jokers in every deck, remember I made the mistake when I wrote that thinking that you had 52 in every deck if you have 2 jokers. That's not true. You have 54 now in every deck if you have jokers. So that means that for every 54 cards, you expect to see 2 jokers. Is that correct? Well, how many 54s go into 1662? Take 1662 and multiply it times 2 over 54. What do you get? What do you get when you multiply 2 times 1662 divided by 54? What do you get? Two times 1662 divided by 54. Actually, that's...
You want us to do that?
You have a calculator, don't you? You don't have to do it by hand. Huh? 61.5. 62? 62 okay with you? All right, so we can round that. It's approximately equal to 62. All right? So I expect to see 62, because what is this? This is the how many jokers I expect. How many jokers do I expect? I expect to see 2 jokers every 54 cards, right? And if I have 1662, then I expect 62 jokers to be in the card. So my expected values here for jokers is 62. So how many what about the rest of these? What do I expect now for all the rest of these? Well, what's 1662 minus 62? 1600. And what do I do with that 1600? Divided by how many? 4. And so it's 400. That wasn't hard, was it? It takes a little bit of thinking. It just takes a little bit of thinking. I wanted you to, if you're confused, I just need you to stop, think about it. It makes perfect sense. There's absolutely no surprise here. All right? It really is just a matter of, now if you never played cards before, you didn't know that there were 54 cards in a deck with jokers, then that might be hard. You might have to do some research. But with some common knowledge or some research, you would be able to figure out some basic math, right? Taking your fraction and multiplying times your total, you'd be able to tell how many jokers you'd expect to see if it was random. Yeah?
So, like, this Carson will be on the test Thursday?
Maybe. It's Wednesday.
We should, 'cause they're-
No, they said it was Thursday. I said Wednesday yesterday.
Can we move it back?
All right, can we get moving on this?
No, sir, I still don't get it.
All right. What don't you get?
I don't get none of it. I don't get how you got none of that.
Okay, I guess,
And I don't understand what the question you asking, like,
all right. I mean, there's nothing wrong with not understanding, but to say that you don't understand any of it, it's not very helpful to me. So what I'm asking you is specifically, you're reading this problem,
Okay, I get what you're saying, so.
and I'd like to know specifically what you don't understand.
So I understood it, and then I understood when they gave me the
Hold on. Let me pause this. There's someone on the phone, and is there someone asking a question? You gotta be quiet. To ask her question so that I can explain to her, and I bet that her question, a lot of you would have if you put some thought into it. So let her ask her question. Go ahead, dear.
Okay, so I understood like where the numbers came in and stuff, but then when they asked the question like they said, could it be equally like stuff? It just wasn't.
All right, say that again. Where did they ask this question?
It said, could it be equally likely, or are these not too much?
Discrepancies too much to be random. Okay, so what they're asking is what I said earlier, right?
Do it sound true or not? Like, do it sound possible?
Right. Is it possible to get these numbers even, like you would expect to get 400, 400, 400, 400 for number one, but you get 404, 420, 400, 376. Is it likely that this is just by random luck that you got these numbers instead of 400, 400, 400, 400? Or is there something, is someone cheating?
Yeah, how could you find that?
Chi-square, right? So you calculate chi-square, like I just showed you to do, and then you compare that number to that table. And if that table critical value is if that chi-square is smaller, equal to or smaller than that critical value, then it's just random luck. If that chi-square is bigger, then someone's cheating.
Wait, you said if it's lower, it's a chance, but if it's bigger, it's so much cheaper.
If it's lower, it's due to random chance. If it's bigger, if the chi-square is bigger than the critical value, then something else is going on. It's not by chance. It's either some error, some mistake, or somebody's cheating, or some other explanation, but it's not. Gentlemen, if I have to ask again that you give these other students the respect that they deserve, there's going to be issues. Yes, ma'am.
Okay, so I get that it's 54 cards in the deck, and then 2 out of 54 is the Joker. But why did you multiply the 2 out of 54?
Why did I multiply? Listen to her question. Why did I multiply 2 out of 54 times 1662? Because, yeah, that's right. Because that's how many jokers I would expect to see in the entire, all the cards that were dealt. What were the number of cards that, where did I get 1662? I added all these together. This is what came out. This is what's added. So out of all the cards that come out, how many would I expect to be jokers? Well, for every 54 cards, I expect 2 of them to be jokers. So if I multiply this times this, that gives me how many jokers I expect to see in the deck. And you do the same. You could do the same for all of these. Yeah.
And then you got the 61.5 and the 62.5.
Well, 61. That's what happens when you get about 61.5. But if you round it off, you can round it up to 62.
Okay.
That, I mean, it's just rounding.
And then you subtracted that, and you were subtracting the jokers from the deck.
So I subtracted jokers from the deck, and so that left me all the aces, all the spades, hearts, diamonds, and clubs. And if they have equal amounts, which I have to assume that they do, then I could take 1600 divided by 4, and that gives me how many I expect to see all in all the suits, right? If it's random, and there's an even distribution. If somebody's cheating, or if there's some error going on, then the numbers aren't going to come out the way they should. Yeah.
So now you want us to, do a problem and see if it's like.
Right. So listen to what she's asking. So now what you're going to do is you have your expected, you have your observed. Now all you got to do is make another column that does what? Calculate chi-square. How do you do that? For each of these, you're going to, for the spades, hearts, diamonds, clubs, and jokers, you're going to take your observed, you're going to minus your expected, you're going to square that, you're going to divide it by the expected, and that's going to give you a number. That number goes here. You do that for each of these. Now there's 5 here. So I'm going to take, after I'm done calculating those numbers, what am I going to do with them? I'm going to add them. That's going to give me my sum. That's the sigma, right? Because remember, this formula requires you to sum all of the values 1, 2, 3, 4, and 5. All 5 values, add them together, that's what you get here. That's your chi-square. That's what equals chi-square. So what you do is you just do observed minus expected, square it, divided by expected, do that 5 times in this case, then add them together, and that's your chi-square.
So wait,
That's it.
The chi-square
so
is the
what's the
final
difference
answer.
between the chi-square?
It's not your final answers. That's the number you're going to use on the table to compare your results to.
What's the difference between the chi-square and the critical value?
Excellent question. So when we look at the critical value, it's that difference. So when we have a chi-square, whatever that chi-square is, that's this number that you're going to add together. You're going to take that number, you're going to compare it to your critical value. Now let me ask you, there's 5 possibilities, right? What's my degree of freedom? 4. Because you have 5 possibilities minus 1, it'd be 4. So in this case, we're not going to use the pink line, we're going to use the green line. All right? And your critical value for this problem is not 7.82, it's actually 9.49. Now how do I know that? Because I know that I have 4 degrees of freedom and your critical value is now 9.49. So 9.49 is your critical value.
So your degree of freedom is how many things you have to tolerate about?
How many different possibilities minus 1.
So
All right?
that wouldn't be for the whole chart, it would be for the first 5.
All right, wait, say that again.
So you said that the degrees is like for the columns, right?
So yeah, so what you do is you look at how many different possibilities you have. You have spades, hearts, diamonds, clubs, or jokers. So you have 5 different possible things. Minus 1, so 5 minus 1 is 4, and that's what you're going to use that's the row. You always use 0.05 for your column. So as soon as you find that critical value, that's the one you want to use. So, gentlemen, I'm trying not to say names, right? I know you're not talking, but you're listening.
I'm listening to you.
No, please. So you have a critical value of 9.49. So now whatever this number is, if this number, this chi-square number, right here in blue, right? After you add these 5 different ones, after you observe, minus expected, you square it, divided by expected, do that 5 times, add them together. You take that number, if that number is smaller than 9.49, then the difference between 82 and 62, 356 and 400, 420 and 400, 404 and 400, that difference is just random luck.
So we do the equation 5 different times?
That's right. You do this 5 times and then add them together.
Okay.
It's just that simple. It's really easy. And you know what? Because you're working this hard, I'll move the test back.
Thank you.
So I'll give you some more practice problems tomorrow. Yeah? Yes, you may. Yes. Say that again. Say again, I can't hear you.
So we always have to use this chart?
Always using this chart, yeah. You're always using the same chart. This chart is standard. All right, so this is a practice problem that I kind of wanted you to go over in case you wanted some more practice problems. Obviously, you'll get credit. It'll be extra credit if you want to try to turn it in. So you have large ear corn. That's a corn cob, right? It has 433 grains. Remember, the grains are the kernels. They tell you that there's 271 271 purple and smooth, and 73 purple and shrunken, 63 yellow and smooth, and 26 yellow and shrunken. And you'll see where they're put in the table in a second, but we put the data in the two columns in the table. This is what you have observed, right? This is the observations. So your hypothesis, the hypothesis you're putting into question, the hypothesis is that if an ear corn was produced by a dihybrid cross, and there it is, involving two pairs of heterozygous genes, resulting in theoretical expected value of 93:3:1, right? So this is what you think is going on. If this is true, then we would expect to see a 93:3:1 ratio. So your objective then is to do a chi-square analysis to find out if indeed the data that you've got, this data, these observations match. Does it fit? Does it fit the hypothesis, the main hypothesis here? So let's take a look at that. So here's your data. And here's the formula you're going to use, right? You're going to take your observed number minus the expected number. You're going to square it. Then you can divide by the expected number. You're going to sum these up. So these are the values you're going to get. You're going to sum them up. This symbol means sum. So again, the entire formula is that chi-square is equal to the sum of the observed number. Notice that there's this symbol, this NO, which means number. They want to emphasize that because we don't use fractions in this. You want to get the observed number minus the expected number, square that, and divide it by the expected number. I keep saying number for a reason. Then you're going to add those together. That's what this symbol means. It means the sum of. Right? Add those together, and that's going to be your chi-square. All right. So when we're looking at these data, so we have observed number of 271. Well, what's your observed ratio? So we're expecting to see a 93:3:1. The concept would be, why don't we just go ahead and use this as our 1, right? So if this was going to be 1, it would be, if this is going to be equal 1, then you're dividing this by what? Well, hopefully you know it's 26. So then, ideally, you would want to divide each of these by 26 to see what is the relationship between these four. What's the ratio between these four? So these would be the ratios, the observed ratios. Now these numbers here, are they really that important? Not really, not to calculate our chi-square, but it is interesting to see that we expect to see 93:3:1. What we do see is 10 to about 3 to 1. So is that close enough a ratio to this ratio for us to say that any error, that any difference between the two can be explained by random events? Well, the way we're going to do that is we're going to go ahead and do chi-square. So to find that, you know what your expected ratio is 9 out of 16, but we don't have 16. We have 433. How do we get 433? We added up 271, 73, 63, and 26. That equals 433. Well, 9/16 of 433 is... Okay, so when we take a look at this number, and we say 433 times 9/16, we expect to see 243. Expected number, 243.56 of them to be purple smooth. And does the decimal make sense? Not really, but you go ahead and keep it if you'd like, or you can round up, whatever you feel comfortable doing. 243.56. You expect to see 81.19 of purple shrunken, etc. These are the expected numbers you'd expect to see. This is what you actually saw. So now we use these numbers and these numbers to come up with our chi-square. So when we look at the data and we go ahead and take the observed minus the expected squared divided by the expected, we get these values. We add them up. We take the sum of that. That's our chi-square. So here we see that we have our chi-square value is 7.79. So if we take a look at this significance of 0.05, so we're going to look at this column, and we look and we say that we need a degree of freedom of 3. Why 3? Because you have total 4 minus 1 is 3. Then we come across and we look at our critical value of 7.82. So our critical value is equal to 7.82. That's where we got this here, using a p-value of 0.05. So when we compare the two, we see that our chi-square of 7.79 is less than 7.82, so we accept the null hypothesis. Which means what? Which means that any difference between the expected and observed is due to randomness. Now, that being said, that also means what? That means that our model, our concept that these are, so our original hypothesis, remember that our original hypothesis was that it was 2 dihybrids with heterozygous genes. And the genes were not linked. We would go ahead and accept this. We would accept this as the truth until further data shows us different. So that's how you do chi-square. Again, yet one more example of how to do it. And we've done some more in class. This is going to be on the test, so you have to be able to do all this in class. For problem 3, it says, a genetics engineers attempted to cross a tiger with a cheetah. She predicted the phenotypic outcome of the traits she was observing to be following a ratio of 4 stripes, then we had spots, then we had both. So there's only 3 phenotypes. So could this be a heterozygote, dominant recessive, 2 heterozygotes, dominant recessive inheritance? Is that what's going on here? How do you know not?
Because
Yeah.
doesn't it say like spots only and stripes only? So if you had only those, it would be
Well, first of all, we only have one trait. It's either spots or stripes or both, right? So it's only one. It's not dihybrid, right? So if we only have one characteristic, unless you have 2 genes controlling that characteristic, which is possible. But if you assume only one gene, then there's only one allele, 2 alleles, right? So it's either spots or stripes. Then the other thing is that when you have, if these were dominant recessive, if mom and dad, let's assume they were heterozygote. If they were, what would we get?
3-to-1 ratio.
We'd get a 3-to-1 ratio, right? If they were both heterozygous, right? And we'd get how many? Stripes or spots. You wouldn't get stripes and spots. To get stripes and spots, what kind of inheritance are you talking about? And so well, let's go over again, and we're going to go into this a little more detail tomorrow. But let's go over it again. What are the three kinds of inheritance we've talked about? We've talked about dominant recessive, and what's the other one? You guys read about it in the chapter. You should have by now. It's been 2 weeks. Say that again. Codominant and incomplete dominance. Now, if you're dealing with dominant recessive, how many different traits do you have? 2 traits, 2 phenotypes, right? You only have 2 phenotypes because you're dealing here with only 1 trait. In dihybrid crosses that we've done earlier in the week, there were 2 traits, weren't there? Green and yellow, tall and short, tall and yellow, wrinkled, whatever, right? But this is only 2 traits, stripes or spots. In incomplete dominance, you'd have how many phenotypes? What is incomplete dominance?
You can like really be like white with black, like it all mixed together. They both show up.
No, both shows as codominant. So this is both. So it's neither, right? So it's either spots, stripes, or neither, right? So what you get is something in between, maybe swirls, right? Which would be kind of cool, actually. So swirl, spots, stripes, or swirls, right? As an example, I mean, I don't know, it's all made up there. With both, what would happen is you get spots and stripes. Does that make sense? All right, so codominant means both show up. Dominant means it shows up if it's present. Recessive means it only shows up if the dominant's not there. Codominant means if they're both there, they both show up. Co-dominance, like co-captains. And incomplete dominance means neither one shows up. Something new shows up. So red and white make pink. Not red with white spots or white with red spots or whatever. Okay? This is like the 3rd time I've gone over it, so I would expect that you guys have written this down, that you put it on index cards, that you've gone over it, so when you do take the test, whether it be tomorrow or next week, you know what the heck you're talking about. You're not going to use the excuse, oh my God, I forgot, or I blanked out, or I got nervous. That wouldn't be an excuse, right? Because it's clear that you need to know that. Am I right? Okay. So stripes only, spots only, or both. All right. So I'm going to go ahead and get rid of this if you don't mind. Oh, great. All right. I wish I could get rid of it faster. Oh wait, I think I might be able to do that if I do this. Ah, that's tricky. All right, spots, stripes only, spots only, or both. What were our predicted values? You know what? Let me put the predicted somewhere else. Our predicted were 4 stripes only, 3 spots only, and 9 both. Okay. And what did we actually get? 50, 41, and 85. Well, how do I compare these two? These are numbers. These are actual numbers, aren't they? This is 4 out of what? 3 out of what? 9 out of what? Because the total here would be 16. So that's 4 out of 16, 3 out of 16, and 9 out of 16. So what I'm looking at here are actually ratios. And if I wanted to find out the numbers so I can compare numbers, because remember, chi-square, we don't use fractions in chi-square. We can't say 50 minus 4/16, right? So we can't do that. We have to come out with a predicted number. That's our predicted ratio. What's our predicted number? We'd have to multiply 4 to 16ths times what? The total. This is exactly what we did with the cards. It's exactly what we've been doing all along, right? We have to take and divide or multiply the predicted value times the total observed. So what was the total observed? 176. So 4/16 times 176, 3/16 times 176, 9/16 times 176. What that's going to get us then is the predicted number. How many do we expect to see? How many stripes do we expect to see out of 176? I know I expect to see 4 out of 16, but how many out of 176?
44.
So 44 here, then what? 33, then 99. For a total of 176, right? We're good with that? Now I see that the two totals are equal, so I'm good with that. So here's my my predicted values. Here are my observed values. So now I can go ahead and try to figure out what chi-square would be. And for chi-square, what I want to do is what? That's right, I'm going to take observed minus expected. I'm going to square it. I'm going to divide it by expected. I'm going to do that for each one, and I'm going to take the sum, right? Because chi-square is the sum of all these, right? Remembering that the formula for chi-square is observed minus expected squared divided by expected, and that's the sum of. Do you remember that? Are we good with that? All right, so if we do that, if we take 44 and if we take 50 minus 44 divided by 44, what do we get?
0.818.
0.818. Or 87, okay. What's the next one?
1.939.
And the last?
1.8.
Keeping the same significant digits, and we'll talk a little bit about that at the end of the year, but not too much. Add these together, what do we get?
4.55.
4.55. 7. Okay. Not rounding, keeping everything the same. So 4.557. And now we have to find their chart. Who can look at their chart? How many degrees of freedom do we have in the chart? 2 degrees of freedom. At 0.05, what's our critical value?
5.99.
So 5.99 is our critical value. Is our chi-square smaller than our critical value? It is, because 4.557 is less than 5.990. Do we agree? Yeah.
Did you say look at the 0.5?
0.05. 0.05. Right? Not the 0.5. I made that mistake when I first showed it to you. Remember, I looked at 0.5 and I corrected and said it's 0.05. That was my bad if you messed that up. Are we good? And that's it. So what do we say? Since it is less, what does that mean? What does all this mean? Their chi-square is less than 5.99. We accept the null hypothesis. Look at how I'm writing this, because this is how I'm going to expect you to write it. I accept the null hypothesis the difference between the expected and the observed is due to random events. Do we agree? That's it. That was not hard. Had I not stopped to explain, I probably could have done that, what, 3 minutes, 5 minutes with a calculator, right?
Yeah.
Okay, so that's kind of the timing I expected that you guys be able to do this.
So, excuse me.
Yeah.
So we say no, their hypothesis wasn't a good fit, or don't even answer that part. Just with the,
What was the question?
it says, according to Chisquare test, was there a significant difference between the results and the predicted?
All right, was there a significant difference between the? No, there was not.
So I was saying like,
So-
just put no, it was not on there, or just put I accepted on my.
You could put both. I'm telling you that that's what that all that you just read. That's what this means,
Okay.
right?
Yeah.
Now, what would you to answer that question that you've just read, right? You'd have to say there is no significant difference between the expected because it's due to random events. Is that okay?
Yeah.
And then you'd have to say the hypothesis that we had that we would get 4 to 3 to 9 is a good fit. It's a good fit. It fits, right? The null hypothesis, I accept that any difference is from our hypothesis to what we actually see is due to random luck. So this hypothesis fits the reality. I'm okay with that.
Okay.
Are we good? Let's look at number 4. Number 4 says in the garden pea, yellow is dominant to green. So yellow is big Y. Green is, green is recessive. Agreed? Yes or no?
Yes.
Yes. Inflated then is dominant to constricted. Does it matter what inflated or constricted is? No. But everybody agree with that so far? All right. And considering both of these traits jointly self-fertilize, dihybrid, so there's self-fertilized dihybrids, the progeny appeared in the following numbers. Here's my issue with the way they write this. It's not very clear in a lot of things, but what it is clear to me is the data tells you exactly what's going on. And if you can identify what's going on from the data, just without even doing any chi-square stuff, you already know what the hypothesis is, or you should know. And a lot of it is told to you just by the numbers. So 193, green, inflated, and the genotype for this would be, if to get green, what would I have to have? Little y. Inflated, I'd have big I and blank, right? I'd have 184. Did they give me the genotype? No, how'd I get the genotype? Based on what I was given, I wrote it. I interpreted what was given to me. 184 yellow constricted. So that means Y blank little I, right? And 556, yellow inflated. So that means big Y blank, I blank, right? And 61, green constricted. Now what I'm about to say, it's going to be very important when we get into pedigrees. So pay very close attention. Based on what you know about sex right now and about meiosis, you should be able to tell me what the genotypes of this parent is, this self-fertilizing plant. Yes, go ahead.
Where the genes are from? Is this supposed to be little I,
Oh,
little
yeah. Thank you. I don't know where it came from. All right. You should be able to tell me what the parent's genotype is.
Big Y, little Y, big S.
Right, exactly. Big Y, little y, big I, little I. Right? How did I know that? How did she know that? Here's how we knew that. All right, somebody have an answer? Go ahead.
Wait, okay. All those genes are shown throughout the plants.
Exactly. If you have, here's the key. I keep telling you this. The homozygous recessive is the key to all genetics problems. If you see a homozygous recessive trait here, what does that mean? How did these children get little I, little I? How did these progeny get little I, little I? From the parents, right? So that means that they had to get two little I's. That means that parent had to have what? At least one little I, right? The parent had to have a little I. Okay, and it's self-fertilizing, so it's only coming from one parent. But even if you had two parents, that means each parent would have to have a what? A little I. And here's another homozygous recessive. Here's another homozygous recessive trait. So now you have two little Y's. What does that tell you? It has to be at least one little Y. And there it is. It has to be. And then you have a big I and a big Y. So that means you have to have a big Y and a big I. So you know what the genotype of the parent is based on the fact that you have these outcomes in the progeny. Are we clear? That logic has to be clear in your mind because that's how you're going to solve these. You're going to see a lot of frustrated children trying to solve these problems, and the core of their issue is they have not paid attention to meiosis, crossing over, what's going on with little I, that these genes come from the parents. They just don't, it's not clicking for them. Mainly, in my opinion, because they're not working. They're not doing the work. Okay? All right. So we have this down. This is our genotype. These are our phenotypes. Now, the question then, are these independently assorting? If they were independently assorting, what ratio would we expect? 9 to 3, 3 to 1. We expect if they're independently assorting that we would get 9 to 3 to 3 to 1. And of course, it's not the order we would expect, though. We'd expect 9 of the biggest, which is here, 9 of the double dominants, right? 9 out of 16. We'd expect to see, of the yellow constricted, we'd expect to see 3 out of 16. 3 out of 16 of the green inflated. And we'd expect to see 1 out of 16 for the green constricted. Right? Why? Because this is the double dominant, right? The double dominant ends up being the greatest number. And then the two that have a dominant, one recessive and one dominant trait, those are going to be 3 each. We've been through this like 50 times, I think, at this point, right? Not really, but we've done this a number of times, haven't we?
Yes.
Yeah, I understand. I understand. It's not, it's about putting it together. That's why we're sitting here doing this together now. That's why the test is not today, right? I know, I pay attention, I do my homework. All right, so here you have 3391, okay. But that tells me the ratio. This is the expected ratio. What about the expected number? How do I get that? Obviously, these are the observed, right? How do I find the expected number? Because chi-square requires me to look at the number. Right, how do I gotta add the total. Somebody do the math? 994. So that's my total. So what do I do? And I got the total. How does that help me find the expected number, though? That's right. I got to multiply that times the ratio. So there's my ratio. If I take 3/16 and I multiply it times 994, that's going to give me how many? Times 994, times 994, times 994. What's the number? What number do I get then? For the first one, I get what? 186. Then I get what? 186. That makes sense, right? And the next one? And the last one? Yeah, it is close, isn't it? 62 is close to 61. 556 is close to 559. No, 193 is 186. I mean, how close are they? Well, that's why we find the difference, right? That's why we do chi-square. We're looking for that difference. What's the difference? What's between the observed and the expected? How close are we? Our gut feeling is that it's close, isn't it? That's my gut feeling when I look at this. It's close. But we like, in science, we like numbers. We like objective. Make things as objective as possible. So chi-square, remembering that it's a sum of and I keep writing it, keep saying it, hopefully it sinks into your minds. Observed minus expected squared divided by expected. We're gonna go ahead and do that. So what's the first one is 193 minus 186 divided by 186. Oh, and of course this is squared. And so what do we get? 0.0 0.0 0.263. All right. And the next one is 184 minus 186. Oh, that's a negative number, but it doesn't matter because we square it. Divided by 186. And what do we get? 0.022. And the next one is 556 minus 559. Oh, another negative number. Again, doesn't matter because we're squaring it. Divided by 559 equals what? 0.0115 or 6? 6. Okay. And here we have 61 minus 62. What's going to be a negative number? Doesn't matter. We're squaring it. And divided by 62 is what? What is it? I heard a lot of mumbling.
0.016.
0.016 again? Wow, that's interesting.
No, that wasn't put up.
That's not right?
The one of
Yes, it is right.
All right. It is.
the one above.
Oh, the one above is not 0.1016?
Yes.
No, that's the one, that's the
Could someone check these numbers? Is it good? I mean, do
They're
it.
the same.
They're the same. Everything's good? All right. I'm just going to go ahead and assume it's right. It doesn't matter, but that's fine. So chi-square is the sum of all these, so we add them all up. And what do we get? Because remember, what does this mean here? What does this sigma mean? Sum. So we're adding them up. Somebody add them up. What do we get? 0.317. That's 0.317. And how many possibilities do we have? We have 4, so how many degrees of freedom? 3. And what's our critical value? 0.05.
7.82.
7.82. So is our chi-square, is 0.317 less than 7.82? Yes, it's very, and in fact, we were really good, weren't we? Our gut feeling was absolutely right, spot on. This is very close to what we'd expect. Because if the two were the same, if the expected and the observed were the same, we'd have a 0 here. Chi-square would be 0, right? But it's 0.317, so that's pretty good. When chi-square is less than 1, we're in a good place. Yeah.
So does that mean that we'll accept the null hypothesis?
Yes, we accept the null hypothesis. Any difference that we see here is due to random error, and we don't have to worry about it. Our theory, our model fits the data very well.
So
Yeah.
so it proves the 9.3.
Yes, that's right. So 9 to 3 to 3 to 1 is proven. And not proven, we accept it. We accept 9 to 3 to 3 to 1 as being a good explanation. It's a good fit.
So whatever the hypothesis is in this scenario, that's the null hypothesis.
Say that again.
So the hypothesis that's like in the scenario that we're doing is the null one?
No. The hypothesis, we're testing that hypothesis. And to test it, we have 2 other hypotheses, 2 possibilities. And those 2 possibilities, we call them either the null or the alternative. So we have this, I'll do it the way the website that I sent you does it, right? So we have this shoe. Oh Lord, I can't draw, so don't make fun of my shoe. I got big feet, all right, so back off. All right, whatever. See, this is every time I draw, I just don't want to draw. I want to just put a box. There. So, does this shoe fit, right? If you have this foot, oh Lord, I have to draw a foot, seriously. It's Baron's feet that have to have hair on it.
My feet, I don't have hair on my toes.
Well, you're the one who started it, so I'm back.
I didn't start it.
I heard your comments, Baron. Yeah, and I'm recording. I'm keeping it on here too. So there.
See my toes.
Stop being creepy. All right. So, anyways, the question is, does the question that you're so this is your foot. Your foot is like the hypothesis, 93, 3 to 1, and your question is, does this shoe fit the foot? Does 9 to 3 to 3 to 1? This is the foot is the data, and the shoe is your 9 to 3 to 3 to 1 explanation. The question is, does this explanation fit this hypothesis or this data? And in order to answer that, you have two possible answers, right? It's either this is a good fit or it's not a good fit, right? All right, so if it's a good fit, so how do we do that? How do we decide if it's a good fit or not? We have the null hypothesis. And that null hypothesis is that there is no difference, so it's a good fit. Any difference is due to random walk. Or you have the alternative hypothesis, which is it's not a good fit. Right? And something else is going on. So we accept in this case the null hypothesis. We accept the null hypothesis, which means that our original hypothesis that we would get 93 to 31, our prediction, our original concept that this was these genes are not linked, they assort independently, right? We accept that because we're accepting the null hypothesis. Any difference from the predicted between the predicted and the actual outcome or our observed outcome is due to random luck, and this concept of independent assortment of genes is a good fit. Yeah.
Kind of weird. Would you have learned this during the 90s if you were in high school?
Would I have learned this?
93 to 1.
93 to 1, yes, was a standard high school. Now, the Chi-Square was something they added on later because they want you to really focus on, there's a good reason to add Chi-Square to this discussion. I don't think Chi-Square, we used to do stuff that's similar to Chi-Square. We used to find our percent error in lab experiments in high school. So in the 90s, when I taught in the 90s, I started teaching in the late 90s, I can tell you that we did. And when I was in high school in the 80s, we would try to find something called percent error, which is similar to. This is a little more advanced, but I think it's a better explanation. And I know that you guys can handle it. It just takes a little more work. And you agree the math is not that difficult, especially if you have a calculator. Now the hard part is what? There's a lot of steps. The hardest part here is remembering what's the expected ratio. What's my theory, right? It's the theory. So this really, if you can apply Chi-Square to the problems you just got today, you got these problems today, yes? Everybody has a copy, those of you that came in late as well? Those people that had those problems, today's problems I expect you to be able to do them. Remember, I'm here during advisory if you need some help. I think we'll have some tutors. I know you had some issues. You texted me at Jupiter, and I just went through the problems now, right? So make sure that that's what I was planning on doing, that's why I didn't answer you right away. Yeah?
If you get a Chi-Square value somehow at 0, does that mean that what it is, like your null hypothesis, is proven to always be true?
Well, it proves that you're right spot on. But I gotta warn you that if you get a Chi-Square at 0, and you have a lot of data, people are gonna wonder what's going on, because they're rare. I mean, really, the idea that you get perfect results is scary. Yeah.
I did the homework, but today I made the corrections because I had a couple errors.
That's right, upload.
So
If you have errors, I haven't graded this assignment yet, so if you have errors or you didn't upload it now. All right?